Good afternoon. Welcome, everyone, to today's conference call. At this time, your lines have been placed on listen only for today's conference until the question and answer portion of our call, at which time you will be prompted to press star one on your touch-tone phone. Please ensure that your line is unmuted and please record your name when prompted so that I may introduce you to ask your question. Our conference is also being recorded, and if you have any objections, you may disconnect at this time. I will now turn the conference over to our host, Ms. Irene Ihir. Ma'am, you may proceed.
Thank you. Hello, I'm Irene Ihir of CDRH's Office of Communication and Education. Welcome to the FDA's 43rd in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to development and validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Timothy.
Wow, 43 town halls to date. That's a lot, and we'll keep going as long as you all think this is helpful. And these sessions are designed to provide assistance to all COVID-19 test developers, whether they be molecular, antigen, and/or serology tests. That's why we do this, and obviously we have a lot of interaction with essentially all developers who submitted a question or an application to the FDA through our office, and those are happening sometimes 24/7. And that's how we do it. I wanted to give an update on authorization. So as of yesterday evening, we had authorized 331 tests and sample collection devices, including 247 molecular tests and sample collection devices, 70 antibody tests, and 14 antigen tests. There are 37 molecular authorizations that can be used for home collected samples. There's one molecular prescription at-home test, one antigen prescription at-home test, and one over-the-counter at-home antigen test. We expect to see more home collection kits come in wider availability for that, including when prescription is not required in that home collection kit, we are calling it direct consumer. And you can search on the FDA authorized website for any direct-to-consumer home collection kits. When it's a test in the home, we're calling it over-the-counter. And when it's not prescription. We expect more collection kits, including more direct-to-consumer kits, and more at-home tests, including more OTC at-home tests. So those are obviously priorities, along with very high throughput central lab tests. So with those brief updates, let's move right into questions, and we look forward to hopefully helping you out today. Thanks.
Thank you. At this time, if you would like to ask a question, please press star one on your touch-tone phone. Please ensure that your line is unmuted, and please record your name when prompted so that I may introduce you to ask your question. Our first question is from Shannon. Your line is open.
Hello, Shannon Clark here with UserWise Consulting. When we conduct home use human factors testing, it's inappropriate to ask lay people or the participants to please read and follow the instructions as we observe them using the product. And since typically participants refer to instructions less than 60% of the time, after they're finished using the product, we typically ask user and manual comprehension questions to assess the understandability of the instructions. So For over-the-counter human factors testing for antigen tests and home-use molecular tests, does the FDA require user manual comprehension testing? Or does the FDA acknowledge their own 2016 human factors guidance indicating that a sample size of 15 participants is adequate for uncovering usability problems with the instructions?
I don't believe we require the questionnaire at the end. Libby, do you know on that, or recommend it? I don't believe that we recommend the questionnaire at the end. I think it's nice to have. It's nice to have if there are any issues in the testing, so that you can drill down to if if any updated instructions are going to be helpful for redoing the study with the modifications to make it work. But I don't know that that questionnaire afterwards is recommended. Bubby, do you have that information handy?
Right. I don't believe we currently have that as a recommendation in the template. We do think that it's important to consider usability and user comprehension, especially for over-the-counter tests when there would be no involvement of a healthcare provider. And that is something that we consider is the appropriateness of the labeling for a lay user. But we do not have a specific recommendation for that questionnaire in the template right now.
Yeah, I find that labeling defects tend to shine through during simulated use anyways, so asking questions after simulated use or actual use doesn't really add too much, but prolongs all the sessions and makes it more costly.
Thank you. That's
okay.
helpful feedback.
Thank you.
Thank you. Next question is from Brian Jones. Your line is open.
Hi, thanks, Tim. So I tried to ask this question last week and was cut off at the end of the call. This is just for some clarification about testing paired saliva and NPS specimens. So the first part of my question is that the EUA template doesn't seem to have any information regarding the investigation of discordant results. And I was wondering if you had any suggestions on how to do that. For example, testing on a separate EUA that was already authorized for use with NP, even if it wasn't already authorized for saliva, if that would be a sufficient investigation.
So the MP swab, when validating oral fluid or saliva, that is a recommendation to use an MP swab. The MP swab, we recommend you use a high sensitivity molecular assay with an extraction in the procedure as the comparator. Now, you could have discordant results between saliva and that MP result. Hopefully minimal, but we've seen it in some cases, and from early days, and for whatever reason, some saliva used in some assays and oral fluids does present a challenge for some assays. So that's why we continue our recommendation for MP swabs. So I think hopefully you're still on the line. You can clarify your question about how to resolve discordances between a comparator test done with the MP swab and the candidate test done with saliva.
So in this particular case, the test that we are validating for saliva already has an EUA for NP. And I've seen that at least in some cases there have been EUAs authorized for manufacturers where what they did is they compared the performance of their test on saliva and then also used their test as the performance for the comparator for NP swabs.
I think that's okay. So what you're saying is
that's what we would plan to do.
If there's discordance, what do you do with the discordance?
Right.
And that would be false negatives in saliva or false positives in saliva, or both.
Yeah, either way, where in both cases, our test, the candidate for saliva, our test is what is being used to test both the saliva and the NP specimen. So I'm proposing that we would use another EUA that was authorized for NP, and we could test those specimens to try to do that discrepancy analysis.
So back to the original question, we may obviously prefer a different test to be used as the NP comparator test than the one you've developed. It's a little bit cleaner that way as far as, a good, it may be in earlier days we didn't have many options to choose from. We may allow them. On the other hand, if we've allowed in the past, and you want to pursue this route, I would get, I would get concurrence by the FDA review team through your interaction, directly with the reviewer or through the template's email address or a pre-EUA. You can use a you can do discrepant resolution. I forget what our minimum recommendations are. Probably if you want to, resolve both positives and negatives, that you do equal numbers of both. The table could be annotated if you do that, which means in the footer you can explain what the alternative method results are. It should be another high sensitivity comparator test with an extraction molecular test. There is a way to do composite results. So the comparator, rather than being a single test, could be 2 or more tests. If you do that for all samples, you can have a composite comparator. There would need to be rules established, like perhaps both have to be positive or only one can be positive. Again, if you're going to go that route, I would double check with our review team. Okay. Hopefully that's helpful, but it's certainly allowed to do resolution of those discrepants, but we would need a minimum number of data points to be able to display that in the footer.
Okay. Thank you.
Mm-hmm.
Thank you. Our next question is from Elaine Allen. Your line is open.
Hi, thank you very much. My question is in relation to the notification process. We followed the guidance and the information online in the notifications for emergency use authorizations, FAQs, for commercial manufacturers distributing serology test kits. Our assays validated. We notified FDA appropriately of our intent to distribute and intent to submit our EUA accordingly. It was our understanding that once we notified FDA and received notification via auto reply that we could begin to distribute. However, FDA replied we could not begin to distribute until the notification was accepted. Would you please provide an update on the notification, acceptance process, and timing? Does FDA ever deny distribution at this stage, or is this a formality to be sure everything is in place?
So from the beginning, you get an email response that says that you had submitted it. It's an automated response. But our team assesses the request and makes sure that some key boxes are checked there to make sure that it's something that we can allow to go up on our notification list. So the official response for allowing notification comes in a subsequent email. We typically try to turn that around pretty quickly. So if it is taking more than 48 hours, 2 business days to get a response, I would ping them again. And I would also, you can let Toby and I know through the template email address, and we can look into a specific situation. hopefully that's helpful.
Yeah, that is very helpful. And I just want to say the response team has been wonderful. I mean, very quick. And I just wanted to place that out there. But we were just a little confused on when we could start to distribute. And so I'm assuming there's one final email that will come in that will say we're good to go.
Yeah, you get an immediate response, and then it goes to a quick review team to make sure that everything's there for proper notification and placement on our notified list.
And just to add to that, because we have had some situations where developers have sent notifications for tests that are not eligible for the notification policy. And so just to clarify for everyone, obviously I don't know which test you're referring to, so I don't know if this applies to you, but just to clarify for everyone listening, one of the areas, especially for serology tests, is home collection with dried blood spots is not, and home collection at all, for both serology and diagnostic tests, is not eligible for the notification policy. So that is one area where we've seen a number of notifications come in that we've had to respond back to the developer to tell them that they cannot distribute yet because that is not under the notification policy.
Oh, okay, good. That doesn't apply to us, so I'm happy to hear that. Oh, I'm sorry. Did I disconnect?
No, you're still
on.
Here.
Okay. I heard a stranger. All right, so they did come back with some comments on what they'd like to see on our IFU. We confirmed that we had made those changes, and
so we don't really want to get into the specifics of any one application. So
no, I understand.
We can answer general questions here, or give you information on how to resolve anything with Toby and I, or with the review team. But we don't want to really get into the details.
Yeah, I understand. I'm sorry about that. Thank you very much for your time. I appreciate it.
Thank you. Our next question is from Eli Railbot. Your line is open.
Yes, hi. I was just wondering regarding getting the tests that a person can take at home, those home tests. I've heard different options that are going to be available either in CVS pharmacies or even that you can just pick up or get sent to your home and buy them on Amazon even. Is there any specific date that will make a big difference for us to get our kids back out and to go back into the public, so to speak, if we can take these home tests so we know where we're standing, at least 90% if it's accurate? Is there a specific date that we know that those are going to be available for just the regular people to be able to pick them in CVS? And if they will, is there a specific brand that we know we have to look for? Thanks.
Yeah, so, the FDA does not develop tests. We review test submissions and look to make sure that it's an accurate test. And if we can authorize it, we'll authorize it. We also can't really tell a developer how they go ahead and commercialize. So once we've authorized an at-home test or a home collection kit, that goes up on our website, and it's really, up to the test developer to figure out how they make that available to patients and consumers. In some cases, the U.S. government funds efforts to make these tests widely available, and you've probably seen some press announcements about that. So again, it's not up to the FDA how these what a given manufacturer does once they get an authorization, as long as they're promoting it appropriately for what we authorize.
Got it. I appreciate that. Is there any on the horizon that on your part is approved, and then up to them how to market it?
All of them are that way, and we've authorized 3 at-home tests. One is over the counter, and 2 are by prescription. And then we've authorized what did I say in the beginning, over 30 or 40 home collection kits. Some of those home collection kits are available direct to consumer already. Again, you can look at the FDA website to see which ones those are. You can reach out to the companies and they will inform you about how you can get a hold of their test or...
Thank you. Okay, I appreciate it. Thanks.
Mm-hmm.
Thank you. Our next question is from Kodamudi Venkat. Your line is open.
Good afternoon. Thank you for taking my call. Many times, it is mentioned that in terms of priority of test reviewing, especially the serology test has a low priority, and the high priorities are the home collection kit and the antigen test and etc. And one of the things, the high priority is high throughput test. Can FDA give a description or a definition what is considered high throughput instead of making some subjective determination? Is there any little bit more insight into what constitutes a high throughput test, please? High throughput is something that can be fluid, and so we haven't posted that information. But you can check through the pre-EUA process or perhaps through a question to the templates email box if your test would qualify as high throughput. As far as the other priorities, serology tests that are point of care, serology tests that are at home, or serology home collection kits, as well as serology high throughput tests, are all high priority tests. I would also say that that neutralization tests, serology neutralization tests, are also currently high priority. We've only authorized one, so when we get any subsequent neutralization assay submissions, we make them all high priority right now. So hopefully that clarifies it. Serology tests in those categories are high priority. Thanks. So the serology test for vaccine differentiation, vaccination versus infection, is it a priority? Or previously you mentioned that it is to go through CBER. Can you give a little bit additional information, please? Yeah, we haven't authorized any tests there, but we would work in conjunction with CBER. And if someone comes in with a complete clinical trial showing, basically efficacy for a test that predicts vaccine, either someone needs a vaccine or somebody has benefited from the vaccine, if they come in with complete data for that, we would try to move that along pretty quickly as well. It's a pretty big ask to show that you can use test information in that way, other than you've developed antibodies or not, which is an easier validation. Thank you so much. Mm-hmm.
Thank you. Our next question is from Kristen Bankert. Your line is open.
Hi, yes. Thank you for taking my question and hosting these town halls. At last week's town hall, a caller had mentioned they were advised during the pre-EUA review that it would be acceptable to use an EUA-authorized multi-analyte test for the detection of flu targets this year. However, beyond this year, it would require a de novo or a 510K. Do you have any updated thinking on this topic? Specifically, will this be required for all multi-analyte tests, as we did not receive this feedback during EUA review? And for the clinical studies needed to support the de novo or 510K conversion, will it be acceptable to use flu retrospective specimens since flu prevalence is low this year?
Thanks for bringing that question up. Actually, we forgot to announce that at the beginning of the call. I think there was some confusion,
there was some confusion with a particular reviewer on that topic, but we've clarified that. So, the bottom line is that I think the topic had spun to what would happen if the emergency declaration ended, rather than if something is EUA authorized, how long could it be on the market, and as long as there's an emergency. I anticipate this emergency lasting years, and we have prior emergencies that have not been undeclared. Ebola and Zika and enterovirus, perhaps among others, are still open emergencies, even though we're years past them. I expect that to be the case for COVID, and in which case there will be no rush by anybody to at least from the FDA to convert these. Now, we are developing a guidance that will guide developers who want to convert, and should an emergency no longer be declared, we'll provide for, we hope, a reasonable timeline for developers to convert their assays from an EUA to a full authorization. So, bottom line is, I believe there was a misunderstanding, and a panel test that's authorized via EUA will, in all likelihood, be able to stay on the market given no problems for as long as the emergency is declared, and can be used not just this season but next season as well. So hopefully that clarifies that.
Yes, thank you very much.
Thank you. Our next question is from Lisa Neibauer. Your line is open, ma'am.
Yes, hi. Thank you very much for taking this call. I just had a question regarding how FDA is viewing the recent studies that compare PCR to culture and specifically show that for CT values over 30, those are really considered a non-infectious sample. So as it pertains to the comparison for antigen to PCR, is it really relevant to have that comparison in populations with a CT over 30 given these studies?
It is important. That's a question that we get frequently. The CDC, APHL, and CAP have all said that CTs cannot be used to determine infectivity. Culture is a notoriously insensitive method. Now, if you're positive via a culture method, then you know that it was intact virus that could potentially infect others. But a negative doesn't mean that. I have not seen any outcome studies relative to this hypothesis, and I think it may not be ethical to put people at risk of contracting the virus just to test a hypothesis like this. I think if you search the literature, you will also see that, and we have data that's not public, that in the first 5 days of symptoms, patients can have very high CTs on any given molecular result, a comparative result, as high as 40, and but well over 30. In fact, we often see up to 25% of the results in the first 5 days of symptoms above 30. Someone who has symptoms in the first 5 days of symptoms and has COVID confirmed by a high specificity, high sensitivity molecular assay probably needs to be deemed potentially contagious. So that's just science, and that's what we're basing our decisions on. Now, that said, we are encouraging of all rapids to come in. Rapid test can get authorized, we've authorized tests that have only been examined in the first 5 days of symptoms. And, and if they perform well on those first 5 days, when people are generally thought to be more infectious, compared to molecular test, which is comparisons made there for accuracy. We want to know, is this patient in the first 5 days of symptoms really have COVID or not? And molecular is the gold standard to tell us that. We are not looking at whether an antigen test is accurate, 2 weeks out or 3 weeks out. So we believe this is a very reasonable recommendation for validation. The other thing is that, we just don't know for sure, although I think there's some data that's creeping out, what the initial, when someone first begins to shed virus, is it really high amounts or could it be really low amounts? And we look forward to seeing what that portends. But we would love to see a direct diagnostic test perform very well in those early days of viral shedding. So hopefully that addresses the question. It may not be what you want to hear, but
yes, thank you. I think the only thing I would add is just that we have seen on the NIH website that there is really not a correlation between CT value and days since symptom onset. So I think it's a little difficult to use days since symptom onset as some sort of a marker. Perhaps consumers can't recall or don't know that number very accurately. So thank you.
I see. But there is the other thing to say about CTs and APHL, American Public Health Labs, CDC and CA, College of American Pathologists, has stated very clearly that you cannot compare CTs between assays, even within the same assay in the same lab, between different technologists, down to that level. The molecular assays that we've authorized, none of them are quantitative. They have not been validated to our knowledge, at least not the ones we've reviewed. We haven't authorized a quantitative molecular assay or a semi-quantitative molecular assay, which require at least 2-point calibration and demonstrating good linearity over the dynamic range of the assay, and at least 2 lots of calibrators and probably 3 lots of reagents, because there can be lot-to-lot variability. You want to know that the calibration can be set across lots and be consistent so that you get accurate quantitative or semi-quantitative data over time with an assay. So we have not authorized a single one of those. I'm not seeing a lot of interest in that, but if you want to be truly quantitative, that's the way to go. The challenge with that is, even if you have a quantitative molecular assay, a very good molecular assay, it's pretty clear to me, at least, and I think many people, that a respiratory sample from a swab, is just not something that is going to be consistent throughout a day or over time. There are other variables when you're doing some sampling the nose, or perhaps even sampling saliva in the mouth. You just can't count on consistent virus shedding through a day or over days of when somebody is infected. It's different from other true quantitative molecular assays, for example, which are blood-based, for, say, HIV, HBV, HCV. I mean, those are excellent assays that have been authorized by the FDA, and they involve a sample type, whole blood often, that is well mixed and is a reliable sample type for determining viral loads and monitoring patients, particular HIV and HCV patients, through therapy. So long-winded answer there, but we get this so often. I wanted to be little bit more complete about our thinking on this topic. Thanks.
Thank you. Thank you very much.
Thank you. Our next question is from Ann Wright. Your line is open, ma'am.
Hello. Thank you for taking up my question. I have a two-part question. One of it, first one is in regards to EUA guidance for antigen. Is the expectation that, once we get the authorization, expectation is before release and distribution, are we supposed to complete our design control portion first prior to, being able to distribute it?
We waive most design control requirements with an EUA authorization. That's pretty standard.
Okay.
We've done that for all of the assays. We require a couple of things. We require you to do MDR reporting and complaint handling.
Okay. After the fact,
is there anything else that we require as far as design controls and quality system requirements?
I believe that's it. The best way to check would be to look at a recent letter of authorization for a test that is similar to yours, and look at what we have waived versus what we've required.
Okay. All right, we'll do that. And sorry.
You had a second part, you said?
Oh, yes. So, just to continue on the first part, just to make it clear. So, if you are kind of basically using it, that you want to potentially use the data for later on for the 510K submission for the future, is it advisable, I guess, to basically, create your DHF and make sure that you have all your data and ready so that you could, be prepared for the future 510K? Would that be advisable?
Yeah, that's an important question, and we've certainly seen a lot of new developers, new at least to the FDA, who may not have FDA-compliant quality systems in place. And the guidance that I mentioned earlier on this call of the transition from EUA to full authorizations will address the quality system requirements as well during that transition. So if you want to transition to a full authorization, it doesn't hurt to start working on that now and get things in place. But look to the guidance once it's made available for more details on that.
Okay, but it's not available yet.
No. Cody, you had something else?
Yeah, just wanted to clarify. I believe that there are 3 portions of 820 that we do not waive. Those would be acceptance activities, non-conforming product, and statistical techniques. So you can take a look at in one of the recent authorizations as well. And also, to what you were asking about, preparing for a 510K or future use, we also certainly don't object to any, tests under EUA being developed under a full compliant quality system. That's certainly preferable, but we do waive certain aspects of it.
Okay. So basically continue on with post-market activities.
Right.
As is upon release. Okay. All right, thank you. And then my next question is, under the stability test and guidance document, it's not clear to me whether the expectation is to also include a test for operating conditions, basically like a room temperature, how the device will, the reagents will operate, make sure that they're, still operating as intended.
If it's a point of care test or it's a home test,
or near,
look to the Flex studies and look to the variables that may impact your test performance. And temperature could be very well do that. But again, you can do that with bench testing. You don't have to do that in the field in a home or in a home situation or in a clinic. Okay.
Okay.
The other thing I should add about quality system regulations, there are some times when we'll add into the letter of authorization some additional quality system requirements that we don't standardly ask for all, necessarily for all EUA authorizations. So there may be some specifics that may be technology-based or may be specific for a particular test that came up during our review. So again, I think Toby's recommendation is great. Look at some recent authorizations that mimic what you're doing for that. And then obviously ask the reviewer when you submit.
Okay. So near patient tests and point of care testing would be kind of considered equivalent in terms of that, right? In terms of the operating conditions evaluation?
Yeah. Look at our templates for whatever technology you have for the point of care, and it'll have a list of sort of suggested, recommended flex studies. But each developer decides which ones apply generally to them. But you can also look at recent authorizations at the instructions for use, and those kind of flex studies, I believe, should be described. They may not show all the results, but they may be described in written forms for the ones that we have authorized. And that would be a good indication of what, if you look at a similar technology to yours, what we would be asking for flex studies.
Okay, thank you so much.
Thank you. Our next question is from Luis Jimenez. Your line is open, sir.
Hello, thank you for having this meeting. I just wanted to ask, in terms of the point of care validation, is the FDA accepting the validation information from outside the U.S., or is there a requirement to do a clinical trial in the U.S., for example, with Canadian data be acceptable?
So we do recommend that point of care studies and home studies are done in the U.S. It's sometimes very challenging to assess how closely settings outside the U.S. would mimic settings inside the U.S. We've clearly seen some developers do, quote, point of care study in central labs with fully trained laboratorians, and that's obviously not. And that may be where point of care testing is done in some countries, but it's not where it's done in our country. So that is our recommendation. If you want to do something different, I would submit a pre-UA and outline what you would like to do and see if it's acceptable to our review team.
In the point of having some type of bridging study, do you have any general recommendations to be able to leverage existing data to bridge it with U.S. centers?
Are you talking about point of care studies?
Yes.
Yeah. So I don't. Bridging studies are done on the same sample, but on different devices, or. So, but it may be that you already have some point of care studies done outside the U.S., and if it, through discussion with the FDA review team, if those are relatively close and mimic what a point of care site would be in the United States, that I'll leave it up to the review team to assess that. But it could be that you add to what you already have. You don't necessarily repeat everything that we would ask for in the U.S. that we would ask for a point of care study. So again, it's situational. The team will want to look into your technology, what you've done, and look at the sites that. And the characteristics of the sites where you did point of care studying to assess that.
Thank you very much.
Thank you. Our next question comes from Raymond Boulay. Your line is open, sir.
Yeah, hi, good afternoon. For a prescription at-home test, would FDA grant authorization if mechanisms were not yet in place to report results back to the ordering healthcare provider?
So if it's by prescription, there's automatically some relationship between the prescriber and the patient. They are somehow connected, right? And the reporting that we recommend, but let me say this. We encourage. I think in usually the labeling of a test that we've authorized in this setting, we do suggest that the home test user relay their results to their clinician and discuss the results with them. But the FDA has no way of enforcing that. And we don't make it a requirement for authorization of point-of-care tests and or home tests, that there is a mechanism prior to authorization of reporting results. We do ask in the templates, what are your plans to allow that reporting? And just so folks on the call know, there is a vast underreporting of point-of-care results in the U.S. and in home use tests. In anywhere in CLIA waive settings or at home. There's just a vast underreporting of that, and that's so important for us to help from a public health perspective to help manage best this pandemic, is to know what the results are from all those point of care tests. So usually as a condition of authorization to be handled post-market, we get agreement from developers to find a way to make our reporting possible. So hopefully that helps. Thanks.
And just to clarify, Tim, I believe you're mostly speaking about reporting to public health authorities. In terms of reporting to the patient and the ordering healthcare provider, if it is a prescription test, we do expect the result to go to the ordering healthcare provider.
So
that could be done by phone. That could be done by conversation, or however the clinician and the patient decide.
Just a quick follow-up question. If, for instance, tests were purchased by a university and were distributed to students under some standing order from a healthcare provider, would it be acceptable for results not being shared with the prescribing healthcare provider for the university? I'm thinking about hundreds, if not thousands, of students doing testing with an at-home test kit based on sort of a global prescription, if that makes sense.
So if you look at the tests that we've authorized, we make there's labeling in there that suggests that results should be reported. If you're in a school setting, I think you want to know, and school wants to know those results, right? So you want to know that you're having them do testing for a reason. So, as far as the details go, I don't know that it's under the purview of the FDA. It's more under the practice of medicine, and that's governed more by state and local rules and laws. So what we ask for prescription tests is that they are prescribed, and then we have our labeling that we authorize in the test for suggestions on what to do. And there are flexibilities there. If someone is doing a blanket clinical order for testing, they can provide information in addition to what information you can get from the test in the test kit, or even the patient and healthcare provider fact sheets that the FDA posts online. A prescriber can, say, hey, if you're positive, do this. If you're negative, do this. Or don't do this, or whatever. So it's really up to the prescriber. Tests by prescription for this very purpose have another mitigation that allows us to have a lower bar for prescription tests because the clinician is involved and can advise their patient, about these things. So if you want to have a greater discussion on what might be acceptable on a college campus in their specific situation, I would suggest you send an email
to the Hep email box and ask for Toby and Tim. And we'll arrange a call at the right level to help you out.
Super. Thanks so much.
Our next question is from Josh Perfecto. Your line is open.
Hi, thanks for taking my call. My question is about the interaction of the asymptomatic and pooling claims for high complexity molecular testing. So if the use is going to be pooled testing of asymptomatic individuals, does that just require the asymptomatic and the pooling claims to be validated as in the template, or would you somehow have to kind of like actually do pooling of your asymptomatic samples when you're validating your asymptomatic testing?
That's getting into the weeds. So we certainly encourage pooling. I think it's a great way to expand testing. It's great if kit manufacturers validate pooling so that their customers don't have to validate anything themselves. They simply follow the pooling instructions and the authorization. Pooling can be launched without prior to an FDA authorization. So developers of the kits can validate pooling, and launch and within Toby 15 business days submit. Rather they have to submit, but they don't have to have a decision on the pooling scheme. They can launch right away. So validate your pooling, submit it to the FDA, then you can launch. With asymptomatic, we do want to see enough asymptomatic to know that the test is accurate. The conjoining of pooling and asymptomatic, because if something's authorized for pooling and there are no limitations, there are some tests, rare tests, that are limited to symptomatic patients only. We haven't given that limitation. We're not objecting to prescriptions for off-label use by clinicians if they want to use a test that's authorized for asymptomatics. But the specific question of asymptomatic pooling, I think that's best interacted with our team. But we're going to look at for your particular test, what are the CT distribution of your asymptomatic population? We're going to look at the CT distribution for your symptomatic population. And we're going to look at what the impact of your pooling scheme is on CTs for positives that are pooled. And we probably can make an assessment about whether the data is adequate to authorize both. It's a little bit hard to prejudge, but once we have the data, or you want to run a clinical trial or a clinical study SOP bias, you can do that as well.
Okay, gotcha. Thank you very much.
Mm-hmm. Mm-hmm.
Our next question is from Laura Ferguson. Your line is open.
Hello. Thank you for taking my call. Laura Ferguson with Delphine Diagnostics, and I have a question with regard to qPCR test kits, qPCR. What is the current thinking with regard to the ability of these tests to detect the new infectious variants, highly infectious variants? Is there a requirement for current tests under EUA review to demonstrate that they can detect the UK and South African and Brazilian variants? And is there any requirement for tests that are already on the market to demonstrate this? That is, would new tests that have demonstrated that they can detect the new variants, are they able to state that to potential customers with regard to other tests that are already on the market that may not have demonstrated their ability to detect these variants?
So, we issued a safety alert around the variants and described 3 tests
that we saw that there was impact or potential impact based on variants. We described that I think in that from the beginning, we've been asking developers to take a look at the sequence in the database and do an in silico analysis. We would specifically call out anything that we know is in the database at a percent above 5%, especially in recent weeks and months, because we see that as potentially even one mutation, if it's at 5% or above, could really degrade performance. And we want to know for new submissions, what the impact on the test is. We would also look to the known key important variants out there, like South African, Brazilian, and UK, and look at and assess performance. I can't prejudge what our teams would say, because there could be simply a limitation that says you can't detect the UK variant, or you can't detect South African. But I think both of those are increasing rapidly in our population, and would probably, if a new submission could not detect those, it would put that submission at risk. I do recommend that, or I do encourage, not necessarily recommend, that all developers consider this, that all developers look at their assays now, but in the development of the assays, that they look at this potentiality. And we know that that assays that target multiple parts of the virus are going to, especially the multiple very conserved parts of the virus, are going to be less impacted going forward by accumulation of more mutations in the virus and variants of importance. We're looking to really share this surveillance activity on mutations with developers. We've been doing it for molecular tests from the very beginning, and we continue to look at variants of relative frequency, and also variants of importance, like the three I mentioned, across all of the EUA authorized molecular tests, because we have all the primer and probe sequences. Those are proprietary sequences that we don't share. But we can run those sequences on a weekly basis through our database and see if there's any tests that might pop that look like there might be a problem. We then assess that risk before reaching out to a given sponsor. Once we assess the risk and think that it's high enough that we want the sponsor involved, we'll reach out to the sponsor and work with the sponsor on that. So we issued the safety communication or alert on variants. When we have new established information about any tests that might be affected by variants or mutations, we will, as soon as possible, update our alert or notification of an additional test or tests that may be impacted. So this is a very relevant, timely thing that we're doing. We've said before that it's very hard to assess the antigen and serology tests, but we are endeavoring in interagency groups to figure out how to do that. But we don't have a solution yet for antigen and serology tests.
Thank you.
This does conclude our question and answer portion of our call. I will now turn our conference back over to our host, Ms. Irene Ihier.